afternoon, everyone, and welcome to the RNA Collaborative Seminar Series. My name is Dr. Laura Keffer-Wilkes, and I'm with the Alberta RNA Research and Training Institute at the University of Lethbridge. And today we have two speakers with us. Our first speaker is Elizabeth Trofimenkoff. I said that wrong, but she is a PhD candidate in Dr. Mark Roussel's lab, also collaborating with Dr. Thakor. And she's here to give her presentation, and I will let her take over.
Thank you for the introduction, Laura. I'm excited to be here and present my master's work with you all. Okay, so my master's work was entitled Mathematical Modeling of EIF5B Mediated Non-Canonical Translation Initiation as a Chemotherapeutic Target. This was, like Laura mentioned, done under the supervision of Dr. Mark Roussel and Dr. Thakor at the University of Lethbridge at the Alberta RNA Research and Training Institute. So in an effort to promote inclusivity and accessibility in my presentations, you will always find the following. So on the left-hand side of the slide, you'll always find the agenda or the outline of what we're going to be going through. And at the bottom, you will always find the main message. So in case there's any connectivity issues, you should still be able to follow along. So glioblastoma is an extremely deadly form of brain cancer. It affects 4 in every 100,000 people globally each year, which results in about 250,000 new cases per year worldwide. Now, incidence rates vary, but the highest can be found in North America, Australia, and Northern and Western Europe. It has a median survival of about 15 months and a 95% mortality rate within 5 years. And although there are therapies available, there is still no effective cure for this disease. So just some general background. Apoptosis is an important form of programmed cell death, and it's important during development and also during the cell regulation cycle. So typically what happens is sometimes a cell, such as the brain cell that you can see here on the right, is impacted by some kind of stress. At this point, the cell has to either overcome the stress or the stress will go away, resulting in cell survival, or the stress will prolong, the cell can no longer survive, and it undergoes apoptosis. Now, there are some proteins that can impact this pathway called anti-apoptotic proteins or inhibitors of apoptosis proteins, and these are called IAPs. And specifically, we're going to be looking at one anti-apoptotic protein, ZIAP, which is the X-linked inhibitor of apoptosis protein, which is expressed via EIF5B under hypoxic stress conditions, which we observe in glioblastoma cells. ZIAP interferes with the caspase cascade in the apoptosis pathways, and like I said, it's overexpressed under hypoxic stress conditions like we see in glioblastoma. Importantly, it's translated using a non-canonical pathway using an internal ribosome entry site in its 5' untranslated region, which we will get to in a moment. Well, before we get to that, I want to point out the canonical or normal translation initiation pathway, and the steps are as follows. First, initiation factors 5, 3, and 1 bind to the 4DS ribosome, which is followed by the ternary complex binding, which is composed of EIF2, the initiator tRNA, and a GTP molecule, which is not shown here. Now, this creates a pre-initiation complex, and that is able to bind to a previously formed mRNA complex here, which is composed of the mRNA, EIF4E, and EIF4G, as well as the poly-A binding protein, which is not shown. After the ternary complex binds, you notice that it's not in the proper conformation. So what needs to happen in order for it to be in the proper conformation is it needs to scan until it finds the start codon, at which point the GTP on EIF2 is hydrolyzed and it is able to dissociate. After this occurs, EIF5B binds to the pre-initiation complex, which is shortly followed by the dissociation of EIF3, 1, and 5, which is then followed by 6DS binding. And it results in this complex here on the right, which I'll explain the significance of in just a second. After this complex is formed, EIF4G and EIF4E can dissociate. And the step that gates initiation and elongation is this important step right here, where EIF5B is able to dissociate from the ribosome. Now, there are two important things I want to point out here. The first one is the ternary complex that is composed of EIF2 and this initiator tRNA. Now, like I said, this EIF2 and the initiator tRNA and the GTP molecule form the canonical ternary complex. However, under stress conditions, the alpha subunit of EIF2 is phosphorylated, and so EIF2 is not available to transport that first tRNA to the ribosome. Instead, under hypoxic stress conditions like we observe in glioblastoma, this EIF2 molecule can be replaced by EIF5B, which we will see in the next slide. The second thing I want to point out is this complex at the bottom, like I mentioned. This is what will be called the RIB5B complex for the rest of this presentation, and it's composed of the normal assembled 40S and 60S ribosome with EIF5B still bound to it. The non-canonical translation initiation pathway that we observe during ZIKV translation initiation, like I said, includes an iris in its 5' UTR, and the steps are as follows. First, EIF3 binds to the 40S ribosome, creating this first pre-initiation complex. Afterwards, it can bind directly to the mRNA, creating this pre-initiation complex. And then we have this non-canonical ternary complex composed of EIF5B, the initiator tRNA, and the GTP molecule not shown here, which can then bind. Now, importantly, there's no scanning in this process because this initiator tRNA is in the proper conformation as soon as it binds. After this, the 60S ribosomal subunit can bind and EIF3 dissociates, and then you'll notice we have the exact same RIB5B complex, which then gates this process between initiation and elongation. EIF5B can dissociate to create our totally formed ribosome. So what did I do in my study? Well, I use mathematical modeling to go through and analyze this mechanism. And so you've seen this process already on the right-hand side where the 40S ribosomal subunit and EIF3 can combine to create the first pre-initiation complex. So what you can do is you can write this as a chemical equation, such as you can see on the left, and I can translate that into a series of differential equations. So for example, if I want to look at the rate of PIK1 over time, the forward rate, so the synthesis of PIK1 can be described as K1, which is the forward rate constant, multiplied by the concentration of the 40S subunit, multiplied by the concentration of EIF3. And the deconstruction of the PIK1 complex can be described as K1, which is the reverse rate constant, multiplied by the concentration of the PIK1 complex. And you can do this for every single species or variable in this model to create your model that you're going to use in order to analyze what's going on. Now, there's another important consideration that we need to account for, and this is called a delay. So this may be a little bit new for some of you, but essentially what a delay is it's the time that it takes for either the RNA polymerase to move from, to clear the promoter region on the DNA, or the ribosome to clear the ribosome binding site, or in the case of the iris, the time that it takes for that iris to become available again so another ribosome can bind. This is what we would call tau1, or that first kind of delay, which is called a clearance delay. Now there's a second kind of delay that we don't consider in this model called a synthesis delay, and this would be the time that it takes for that mRNA to be completely transcribed or for that protein to be completely synthesized. So, glioblastoma is an extremely aggressive and deadly form of cancer, and although other therapies are in progress, there's still no effective cure. So this is where my project comes into play. The first thing I did is I modeled the non-canonical translation initiation pathway, and then I determined plausible therapeutic targets. I applied inhibition mechanisms that related to these plausible therapeutic targets to that mechanism, and I determined inhibitor properties that were necessary for inhibition. Then I went back and asked myself, what would happen to the canonically translated proteins if I applied these inhibition mechanisms? And so that's exactly what I did. I applied the inhibition mechanisms to the canonical model and did the same thing. I established inhibitor properties that were necessary for inhibition that would affect that canonical translation initiation. And then at the end of this, I compared the properties of these inhibitors because we want to have a manageable toxicity to the patient. We don't want to damage the patient any more than what is going to happen with the chemotherapy in the first place. So the objectives and research questions here were as follows. What concentration of EIF5B is necessary to inhibit the non-canonical mechanism? What kinds of inhibitors are required to inhibit this pathway? And what properties of these inhibitors do we need for efficient inhibition with minimal negative effects on the patient? So this was the first trial that I did. I looked at the number or percent of ZIAP RIB5B complexes as I varied the concentration of EIF5B in the cell. Now, at normal cellular conditions, the concentration of EIF5B is about 0.1 micromolar. Now, you'll notice that this region here is in the saturated region where we have 100% of possible ZIAP RIB5B complexes. And you'll notice that we have to decrease this concentration quite a bit in order to get into this region where the number of or percent of ZIAP RIB5B complexes is sensitive to changes in the EIF5B concentration. In fact, in order to reach about 50% of the ZIAP RIB5B complexes, we need to inhibit EIF5B by about 99%. So next, I looked at the different modes of inhibition that we could implement into this non-canonical mechanism. The first one is obvious. Why don't we just inhibit EIF5B in the first place? So if we inhibit EIF5B, it cannot create that ternary complex, which means that we cannot deliver that first initiator tRNA to the ribosome, thus completely eliminating this chance of ZIAP synthesis right in initiation. Now, along the same vein, we could think about inhibiting that ternary complex as a whole. So not just EIF5B, but after EIF5B has bound to that initiator tRNA, we could think about inhibiting that complex as a whole so that it cannot bind to the pre-initiation complex. Now, we could think about that in an opposite view, where we could inhibit that pre-initiation complex interface so that the ternary complex cannot bind. And finally, we could think about inhibiting the RIB5B complex so that EIF5B cannot dissociate. This would result in an accumulation of pre-initiation complexes that will not move into the elongation phase of translation. So what are the results of this? For the EIF5B inhibitor, I used a different kind of graphing technique that produced a 3D surface plot. So I'll walk through this a little bit. A 3D surface plot looks at this, in this case, the number of ZIAP RIB5B complexes. So we had a saturation point of 700 complexes. And I'm going to compare that to the changes in the inhibitor concentration and the KD of this hypothetical inhibitor. Now, note that these two axes are on a log-based scale, so you can't get immediate results after that off of this graph. You have to do a little bit of interpreting. Now, in addition, when you look at this graph, you see that there's a relatively large saturation point at the top as far as the parameter regions for the inhibitor and the KD go. There's a large parameter region where we have a saturation of RIB5B complexes. And there's a smaller region down at the bottom where as we increase the concentration of the inhibitor and decrease the KD, which means that we're increasing the affinity of this inhibitor for its target, we have a lower number of RIB5B complexes. And while that's what we expect, we have a little bit of trouble actually getting useful information off of this graph. And so what we can do instead is translate this graph into a contour plot using slices of this 3D surface plot and actually plotting them in almost a two-dimensional way. So this is the contour plot for the exact same 3D surface plot over here. As you can see, this black line here at the bottom represents 700 RIB5B complexes. So this is one of the spots up here at the top where we see the saturation of RIB5B complexes. Conversely, we have this red line here that represents one or minimal RIB5B complexes. So this would give you a parameter range at which we have completely minimized the number of RIB5B complexes. Now, typically what we look at when we're doing cancer studies is the inhibitor concentration necessary to limit the number of complexes by 50%. And so in this case, that's what I did. And that happens to be this second line here, where we have 350 complexes, which is 50% of the total, which is 700. Now, if I arbitrarily choose a inhibitor strength, so I'm going to choose 1 nanomolar, which is a fairly strong inhibitor, we'll see that the IC50 value is about 2 micromolar. Now, I did the exact same processes for the pre-initiation complex inhibitor and the ternary complex inhibitor. The 3D surface plots look very similar, and the contour plots also look fairly similar. And we find that the IC50 values for the pre-initiation complex and ternary complex inhibitors are about the same at 5 micromolar when we assume that the KD of the inhibitor is 1 nanomolar. What's interesting is that the RIB5B inhibitor produces a large region, a large parameter region, where the number of RIB5B complexes is minimized. Whereas when we look at the saturation point at the top, it's this region of saturation is much smaller. And this is further explained in the contour plot here on the right. In fact, when I look at the 1 nanomolar inhibitor and I want to identify the inhibitor concentration at that point, it's off the graph. So I have to do additional calculations in order to identify that. And it happens to be 0.003 micromolar. And that would be extremely promising. So as you can see here, just as an overview, the IC50 values for the EIF5B, PIC2, which is the pre-initiation complex inhibitor, and ternary complex inhibitors are all fairly comparable. However, the RIB5B inhibitor is at least 2 orders of magnitude smaller. And that would be promising. However, I have to point out that this RIB5B complex is both in the canonical and non-canonical pathways. So hence having to go back and check the effects on the canonical mechanism. Going into the canonical mechanism, I considered 2 conditions. The first one was hypoxic conditions, which would be either within the cancer cell or within near proximity to the cancer cell. And this assumed that we have limited availability of EIF2 to deliver that initiator tRNA to the ribosome. So this, if we go back to the graph that we saw at the beginning, would be around the glioblastoma cell. I also considered unstressed conditions. So this would be healthy cells. EIF2 is readily available. So this would be somewhere else in the brain or somewhere else in the body. In the case of hypoxic conditions, we would consider this to be a therapy that would be applied either in like a tumor resection surgery. And in the unstressed conditions, this would be provided either some kind of oral medication or through an intravenous medication, which is why we need to consider the effects on the canonical mechanism. We don't want to affect any healthy cells along the way. I also considered 2 different kinds of proteins, type 1 collagen and beta-actin. They're both very important proteins in our body. Type 1 collagen is involved in the maintenance of wound healing, blood vessel development, muscle and bone strength. And beta-actin is essential for cell growth, cell cycle regulation, and maintenance of the blood-brain barrier. Now, I chose these 2 proteins because they differ in an order of magnitude as to how many mRNAs they have per cell. So I wanted to check to see the effects of these inhibitors based on the number of mRNAs, the number of prevalence of these mRNAs in cell. So under hypoxic conditions, when I do the EIF5B inhibition, I get actually a very similar result for type 1 collagen and beta-actin. I'm just going to show you the type 1 collagen results because the beta-actin ones are essentially exactly the same. And we get a very similar 3D surface plot and a very similar contour plot. And in fact, when I look at the KD of the inhibitor at 1 nanomolar, the concentration of the inhibitor is 3 micromolar, which is very similar to what we observed in the non-canonical mechanism. Now, when it came to the pre-initiation complex inhibitor and the ternary complex inhibitor, which I'll mention in a second, I had to think a little bit harder about how this would implement or affect the canonical mechanism, because the pre-initiation complex is different in the canonical mechanism than it is in the non-canonical mechanism. So we can consider a few different types of inhibitors. First, we could think, if we're looking top down at this interface, we could consider inhibiting the entire pre-initiation complex interface, which would be this option right here. We could think about just inhibiting the spot where the initiator tRNA binds, or we could consider inhibiting the place on the pre-initiation complex where EIF5B would bind. And it turns out that this option here, option C, is the only one that would cross over into the canonical mechanism, because if we're just inhibiting that place where the tRNA binds, obviously that will impact the canonical mechanism as well. And we get a very similar IC50 value than we saw in the non-canonical mechanism at about 2 micromolar. Now, in the same vein, if we're thinking about the ternary complex inhibition, you would say, oh, Elizabeth, the ternary complex is completely different in the non-canonical mechanism, so this should be a safe option. And I would say, I absolutely agree. However, if we have poor specificity, meaning that this inhibitor isn't super specific in where it's actually inhibiting this complex, it could cross over to the canonical mechanism. If we inhibit EIF5B after it's bound to the initiator tRNA, then of course, that's not going to impact the canonical mechanism. The same goes for if we inhibit that ternary complex as a whole. However, if we just inhibit the bottom part of that initiator tRNA where it will bind to the pre-initiation complex, that will cross over. So that's what I used in my study. And I get, once again, a very similar IC50 value of about 3 micromolar. Now, here's what I was worried about. The RIB5B inhibitor looked really promising in the non-canonical model. However, like I said, it would probably impact the canonical model, and it absolutely does. The region at which the number of RIB5B, collagen RIB5B complexes is minimized at a very similar parameter region. And we see the same thing in the contour plot as well. So the RIB5B inhibitor is not a good option moving forward. So in total, when I'm looking under hypoxic conditions at these two proteins, I get very similar results to what I observed in the non-canonical inhibition trials. What was interesting is when I looked at the results of the unstressed conditions. And so this was the result for the pre-initiation complex inhibitor, and it's very similar for the ternary complex inhibitor as well. So if I look at the number of collagen complexes, this region at which the number of complexes is maximized is much larger than it was under hypoxic conditions, and that's really favorable. In fact, when we look here at the minimum number of RIB5B complexes, this parameter region is extremely small. So we're only going to notice this minimum or this drastic effect on the number of collagen and beta-actin RIB5B complexes at an extremely high concentration of inhibitor and an extremely low value of a KD, meaning that we have an extremely strong inhibitor and a really high concentration of it. And we notice that same trend for the pre-initiation complex and the ternary complex inhibitors. So as an overview, the concentration of the EIF5B inhibitor that's required to reduce the number of RIB5B complexes either in ZIAP, collagen, or beta-actin under any conditions is very similar across the board. The same with the RIB5B complexes. The difference occurs when we look at the pre-initiation complex and ternary complex inhibitors at under unstressed conditions, where this could be a promising therapeutic target just based on the fact that the concentration required to inhibit the number of, or to limit the number of RIB5B complexes by 50% is much greater than what we observed under hypoxic conditions for ZIAP. And so here we go. What concentration of EIF5B was necessary to inhibit the non-canonical mechanism? Well, we needed about 99% inhibition. What kinds of inhibitors were required? We said that the ternary complex inhibitor will probably be best. However, there is evidence to suggest that that EIF5B is not essential for canonical translation initiation. And therefore, as a caveat in here, I'm also going to say that an EIF5B inhibitor is possible moving forward. What properties of the inhibitors are necessary? Well, we discussed a number of IC50 values throughout this as well. So Like I mentioned, it is possible for canonical translation initiation to occur without EIF5B. So it can be EIF5B independent, albeit this process occurs much slower. So I would argue that the EIF5B inhibitor or the ternary complex inhibitors moving forward are our best options. The worst inhibitor option is RIB5B by far, and you'll notice that I don't have the pre-initiation complex listed as either good or bad, and this is because while it looks promising, perhaps based on the calculations, the pre-initiation complex is still a pre-initiation complex, and it can still be present in both the canonical and non-canonical translation initiation mechanisms, so I'm hesitant to write that as a good option. In the future, I need to do some EIF5B independent canonical translation trials. So I'm going to include a separate pathway in addition to the pathway that I already have, where the 40S and 60S ribosomal subunits can combine without the presence of EIF5B. I want to mention that this is just one project in a team of 5 groups working on this as in total. The in silico trials that will look at the inhibitor specificity will be performed in Dr. Wetmore's group, and the in vitro and in vivo trials of these potential therapies will be performed in Dr. Thakor and Dr. Jafarnejad's labs as well. I would like to thank my co-supervisors on this project, Professor Roussel and Professor Thakor. I would also like to thank Professor Wetmore, who has been a huge inspiration and a lot of help throughout my academic career. I'd like to thank all my friends and family for the support over the years, the Roussel members for all the intriguing conversations and learning opportunities, all of my cats for the emotional support over the years, and of course, the New Frontiers in Research Fund, which funded this research. Thank you very much.
Thank you, Elizabeth. So we have only a few minutes for question and answers. I urge everyone to use the question and answer option. It's separate from the webinar chat. So please put your questions in there and then I can read them for Elizabeth and she'll actually be able to answer them after she's finished her talk and we move on to our next speaker. So I have one from Nils Walter here. Is it possible that some biological inhibitors of translation initiation, such as the MI-RISC, also target EIF5B?
Oh, that's interesting. I have not looked at that. We're looking at so what I was doing is more of like a theoretical approach of what target could we use or what target could we target, I guess, in the mechanism. I haven't looked at specific molecules, RNAs, or anything that has been synthesized or is readily naturally available to actually apply to this. That's the next step of the project.
Awesome. Why did you choose ZIKV over any other anti-apoptotic proteins?
Yeah, so ZIKV is one, really well studied and really well characterized. It happens to be synthesized using this non-canonical pathway in under hypoxic stress conditions, and it's involved in glioblastoma cell survival. So all of those things combined kind of made it a perfect candidate to study.
Nice. I don't do any computational stuff. I'm mostly a wet lab person. So how long do all of these simulations actually take you to do?
So it depends on what I was simulating. If I was just doing like the normal simulations without any kind of inhibitor, it would take anywhere between like 5 to 10 minutes per simulation. As soon as I got into the inhibitors, obviously that increased the number of equations that I was using. And so those trials would sometimes take at most about 2 hours, but certainly nothing in the same extent of like a week or something. Yeah, so anywhere between like 5 minutes to the 2 hour mark is pretty typical for these simulations.
Awesome. That was great. I really appreciate you taking the time out of your day to answer questions and talk about your research. And I think we'll move on to our next speaker. Thanks, Elizabeth.
Thank you.
All right. We have Darren Gemmill, who is with the Laboratory of Medicinal Biophysics, also at the University of Lethbridge, and is part of the Alberta RNA Research and Training Institute, and under the supervision of Dr. Trushar Patel. So take it away, Darren.
Okay. Can you all see my screen? Yes. Laura?
Yes.
Okay, great. Okay. Hey everybody. So yeah, as Laura was saying, also thanks for having me on with RNA Collaborative Seminar. I'm pretty excited to be here. So my name is Darren Gemmill. I'm a PhD candidate in the Laboratory of Medicinal Biophysics, which is part of the Alberta RNA Research and Training Institute at the University of Lethbridge. And my supervisor is Dr. Trushar Patel. And today I'm going to be talking about an interesting protein, nucleolin, and its interaction with ZIKV viral RNA terminal regions. And we're also going to look at how nucleolin could modulate virion assembly of Zika virus. So now the thing is Flaviviruses are a large family of viruses that are spread across the entire globe, I think as everywhere essentially except for Antarctica, right? And it's pretty much on every continent. Essentially the virus that infects people the most is dengue virus, and which causes on average about 400,000 cases a year. Collectively with all of the viruses, it usually causes about, it's in the tens of thousands of deaths per year. And so flaviviruses cause a huge burden on the planet, to socioeconomics, to the healthcare systems in various countries. And what's unfortunate is if we look specifically at Zika virus and dengue virus, we haven't been able to develop a vaccine for it due to something called antibody-dependent enhancement of infection. And so essentially what happens is if you get infected with dengue virus or Zika virus, or if you take the vaccine, you develop antibodies against it. But when you're challenged again, let's say in the future with dengue virus, dengue is able to use those antibodies to enhance its infection, and it is deadly. It causes hemorrhagic fever and death. And so vaccinating against Zika virus currently is not possible. And so we need to figure out other methods to target the virus, Zika and dengue, but specifically today, I'm going to be focusing on Zika. So now the flaviviral genome can be broken down into 3 structural genes and 7 non-structural genes, which also includes the NS5 gene, which is the RNA-dependent RNA polymerase for the virus. Now, interestingly, the 5' and 3' terminal regions of the virus are non-coding. They are fairly large non-coding RNA. The 5' is typically around 120 nucleotides. The 3' terminal region, depending on the flavivirus, which I'll get into that, can be between 400 all the way up to 800 nucleotides long. What's very interesting is that these terminal regions are highly conserved across all flaviviruses and are critical for viral infection. Now, if you look over here at this RNA secondary structure schematic, the 3' terminal region does vary depending on the species of virus. This region of the 3' terminal region with these 2 stem loops / pseudoknots, this is actually dependent on, it ranges with viral species, but it's essentially dependent on the host that it, or the vector that it resides in. And so whether it's in a different species of mosquito or different species of tick, you're going to have variation in these secondary structures. What's interesting is that the rest of the genes, structural genes, non-structural genes, don't really have a lot of differences. But what we see major differences in these 2 regions on this non-coding RNA, which is pretty interesting. And then also the second region is kind of an RNA-RNA interaction. region, it also can form pseudoknots, which are RNase resistant and allow this RNA to accumulate in the cells. It's been hypothesized that these regions act kind of as a protein sponge in the cell, where these RNAs accumulate in the cell, essentially interacting with human proteins, allowing for downregulation, or I guess suppression of, let's say, host mRNA turnover, splicing. This region actually helps with the cyclization of the genome, which I'll get into that with this region as well. This region is actually the most conserved region out of the entire flaviviral family, this 3' stem loop. This 3' stem loop, along with the stem loop A in the 5' terminal region, will actually coordinate and base pair to each other. And what that does is that allows the virus to completely regulate transcription and translation. So essentially, let's say you have the ribosome binding to the 5' end and the RNA-dependent RNA polymerase binding to the 3' end and then synthesizing 5' to 3', you're going to get a collision, right? If you have translation happening and transcription happening on the other end. So how the virus gets past this issue is by cyclization, where it actually, when it cyclizes, it inhibits the ribosome from binding and allows transcription to happen. And then it can linearize itself, which can inhibit transcription and allow translation to occur. And so we want to study these regions, specifically the terminal regions in general, but this 3' stem loop and this 5' region, the stem loop A, are absolutely, like, if any mutations occur on this, it is lethal to the virus. And so we want to investigate maybe what human proteins are interacting with it, maybe what RNAs, human, let's say non-coding RNA, are interacting with it, and then potentially use these as an exploitative target to inhibit viral replication, because these are so critical. So what I did was, the human proteins that interact with these terminal regions haven't been identified yet. And so what I did was I performed a co-immunoprecipitation pull-down assay with the 5' and the 3' terminal regions. And so again, they were labeled, and I used an antibody in order to capture the RNA. And as well, in tandem, I grew SHSY5Y human neuronal cells. The reason why I use this cell line is because flaviviruses tend to be neuroinvasive. They cause a lot of neurological damage, whether it's central or peripheral. And so focusing in on the nervous system and nerve cells, we deemed it as very important because, you know, coming out of infections and having neurological issues is a problem. And so we wanted to investigate this. And so performing this immunoprecipitation pull-down assay, I was able to identify several human proteins that were interacting with these terminal regions. And as you can see down here, this is just a little schematic, or a general percentage of some of the pathways that these proteins are involved in that the terminal regions were able to capture. And so as you can see, because this is neuronal specific, we can see, you know, Huntington's disease, Parkinson's disease, Alzheimer's disease. Also interesting is we see like mRNA splicing happening, like hijacking of mRNA splicing, which is also fairly interesting. But specifically for this talk today, we're going to be focusing on nucleolin. So nucleolin was identified in the pull-down assay several times, actually in every pull-down assay. I ran this in quadruplicate. It was identified every single time. And so the only thing is that in order for us to do in vitro studies, I wanted to be sure we could characterize and understand the structure and the chemistry of this interaction with viral RNA. So I designed a truncated version of nucleolin. And again, I'll just backtrack that nucleolin is a multifunctional protein. It aids in ribosome biogenesis. It's involved in DNA damage response. it is able to go through, create condensates or go through phase separation when it interacts with RNA and DNA. And so this is actually a critical protein for the cell. Now, so I designed this truncated construct of with just the RNA recognition motifs. I purified it to homogeneity. Here's just a size exclusion chromatogram of the protein, along with a SDS page to confirm purity. I then took this protein and ran size exclusion chromatography coupled with multi-angle light scattering. And what that did was it allowed me to calculate a molecular weight of this protein in solution. And then I can compare that with the theoretical molecular weight of this protein. So this information will tell me, okay, is my protein a monomer? Does it have contaminants? Does it form trimers, etc.? And this does agree that is very close with the theoretical molecular weight. And not only that, it is not as globular as you would think some proteins were, like BSA. So we also calculated using the dynamic light scattering portion of this machine to calculate a hydrodynamic radius. And so the size of this protein in solution, the radius of it was 3.5 nanometers. Also, to further biophysically characterize this protein, we sent it for small angle X-ray scattering, where we used an electron pair distribution function to calculate this protein envelope. So it's just a very rough, low resolution image of this protein, but it's in solution. And so this does provide a lot of physiological relevance with seeing this in solution. And so after this characterization, I went forward into the characterization of its kinetics or binding with the Zika terminal regions. Because, if we do these pull down assays, we're going to identify a bunch of proteins. Some of them are going to be direct interactions. Some of them are going to be indirect interactions, right? If they're forming like, quaternary structures or like heterodimers with other proteins. We just want to make sure that if we're doing this pull down assay and we're identifying proteins interacting with the RNA, we need to make sure that it's directly interacting with it. So performing these kinetic studies is important for understanding if it directly interacted. And so I ran microscale thermophoresis, which is a biophysical technique where you essentially have your RNA labeled, and using a serial dilution of protein, you put it into multiple tubes, and it measures the fluorescence as you provide infrared light to your sample, and thus providing heat. And so essentially measures fluorescence based on the diffusion away from the heat source. And based on this diffusion difference based on time, you can actually calculate a dissociation constant off of this because you get a change in shape, a change in size when there's a complex forming. And so with the Zika 5' terminal region, we actually calculated, or we determined that it had a KD of 640 nanomolar. With the 3' terminal region, 361 nanomolar. And what's interesting though, is if we go back to this 3' stem loop that I talked about earlier, I currently have submitted a paper and it's under revision showing that, and this is actually Zika specific, that this 3' stem loop can actually, a portion of it can actually shift into a G quadruplex, which for some of us that research RNA, that's and looking at G quadruplexes, that is pretty significant. We're trying to understand why these structures are here. Is it for genome stability? Is it to hijack proteins? We don't currently know, but I identified that there is a G quadruplex in this 3' stem loop. And so I wanted to just take this stem loop and see about measuring the kinetics of this. And so because there's previous literature showing that Nucleolin discriminates drastically between long loop and short loop quadruplexes. If anyone's familiar with quadruplexes, you have these spaces of guanines, and these spaces in between the guanines kind of determine the loops of the quadruplex as it forms. And Nucleolin can discriminate or have varying binding kinetics based on the loop size of the quadruplex. And so I ran the 3' stem loop as well, and of Zika virus. and determined that it had a KD of 174 nanomolar, which was pretty interesting. And so now what we also wanted to see was how does nucleolin potentially a nucleolin knockdown impact viral infection? So with collaborators in Quebec with Dr. Laurent's lab, they ran plaque assays essentially, and knockdowns of nucleolin occurred during Zika infection, and we see there is a decrease in plaque forming units when we knock down nucleolin during Zika infection, and when nucleolin is knocked down in dengue, we also see a decrease in plaque forming units. But what's really interesting here is when you look at knockdown of nucleolin, and then you do qPCR after to look at the amount of viral RNA transcripts in the cell, when you knock down nucleolin, we see a substantial increase in viral transcripts, which is really interesting. And also, you can see in dengue virus, when you knock down nucleolin, a huge increase in viral RNA transcripts occur. Now, this kind of leads to two hypotheses kind of being like, why is this happening, right? So hypothesis one is that with this plaque assay, nucleolin helps virion morphogenesis or the early steps of viral packaging, right? So, or hypothesis two is that nucleolin acts as a pro-human factor, negatively modulating viral replication. So as you can see, maybe when nucleolin is not knocked down, you could see a decrease in viral transcripts, versus if nucleolin is locked down, you see an increase in it, right? Or are we seeing that if nucleolin is knocked down, are you seeing that less functional virions are being formed, and so we get this accumulation of viral transcripts in the cell? And so we wanted to also confirm this on our side, and so we did cell culturing as well, but using a Zika replicon system where the capsid protein is actually knocked out and replaced with a luciferase reporter gene. And so what we wanted to see here was, okay, is nucleolin acting as a pro-human factor, negatively modulating viral replication, right? And what we can see is that when you knock down nucleolin, essentially there's no difference between the amount of viral translation and transcription that is occurring in the cell. And so we tended to side with the hypothesis one, that nucleolin is actually helping virion morphogenesis or the early steps of viral packaging, right? So you have like the capsid protein, you have the viral RNA, and this is what we're trying to characterize and understand is, is nucleolin kind of allowing this interaction to happen, and how is it doing it, right? So now, again, with our collaborators in Quebec, so nucleolin, we find that it is that it does subcellular localization. So when you look at the cell culturing that they did and did immunofluorescence, you can see the control right here where they added DAPI, where you can see AT-rich DNA, which is, you can see it localized in the nucleus. You can see as well, nucleolin, it's primarily located inside the nucleus, right? And then as a control, NS3, which NS3 is a non-structural protein of Zika or Flaviviruses in general. And so obviously that's not going to be present. And then this is the merged data. Now, when you look at Zika viral infection, again, DAPI, but then you see with nucleolin that it actually localizes outside of the nucleus as well. And so it's kind of in these, you can see kind of these packets or pockets, right? And what this has been described as is replication factories. And then when you couple it with the NS3 protein, which NS3 is actually a critical protein for viral replication. It's a serine protease. It interacts with RNA. It significantly helps with just the entire process of essentially translation. So when the structural, non-structural proteins are being made, NS3 can come in and essentially cleave and make functional viral proteins and assist with packaging, right? And so that's also being localized outside of the nucleus, where nucleolin is too. And so, and then here's the merge data. And then we're also seeing the same thing with dengue virus. Dengue virus, we see these replication factories. Nucleolin is co-localizing outside of the nucleus. And then, yeah, merge data. So yeah, it's, again, this is very preliminary data. We still need to answer quite a few questions. Again, if we wanted to confirm if the interaction between nucleolin and the RNA is happening, we would perform something like FISH. And so now what we know so far is we know that nucleolin is exported outside of the nucleus during viral infection, but we don't know how that's occurring, right? Now, the RGG domain has been shown to control nucleolin trafficking outside, but also the trafficking happens by phosphorylation, right, or sumolation as well. So when nucleolin is phosphorylated or dephosphorylated, that essentially predicts or allows it to be transported outside or inside the nucleus. We don't know that mechanism. Also, are there any viral or human components mediating this interaction at all? We don't know. These are all questions we have to answer for the future, right? Now, the thing is that the NS2B and NS3 complex, right, you have this protease as well, you have this protein. This, as again, NS3 is multifunctional, right? It interacts with RNA. It's a serine protease. So is this maybe mediating the nucleolin RNA interaction, right? And then finally, is nucleolin driving the RNA to the assembly complex? So now the thing is that previous literature has shown that dengue virus and the dengue capsid interacts with nucleolin. And so that's also why we're hypothesizing, well, maybe nucleolin is allowing this interaction with, it's interacting with the capsid, interacting with the viral RNA, and then kind of coordinating this packaging, right? And so we have to investigate this more. Also, we don't know if the genome, if it's cyclized during packaging or if it's linear during viral packaging. Maybe nucleolin plays a part in that. Maybe NS3 plays a part of that. We're not sure. Also, you saw with those immunofluorescence pictures that you kind of saw these replication factories. Now, these replication factories are also hypothesized to be cytosolic condensates. Now, what's interesting is that nucleolin forms or performs phase separation or can form condensates in the cell, right? Now, is nucleolin maybe helping with that? Allowing this local environment of viral replication to happen, which is a significant help for viral infection, right? If these are just floating around the cytosol, it's hard to localize everything, right? So, but if you form a condensate or do phase separation, then you can have this replication factory and everything in proximity. And also maybe that's providing immune evasion, right? Having these factories. So again, many questions we still have to answer, but it's a pretty cool study nonetheless. So just in general, I'd like to thank my entire lab for their help with this project, especially Igor and Kadir, my supervisor, Dr. Trushar Patel, and our collaborators as well, Dr. Laurent and Clement. And yeah, that's it. Thank you very much.
Thanks, Darren. That was really great. And I just encourage everyone in the audience, if you have a question, to again use the question and answer chat feature. I do have a question. And do you have a specific reason of why you use that poly-C as a negative control for your binding affinity studies?
Yeah, so now nucleolin tends to interact with AU-rich DNA or /RNA because you have uridines, but essentially it interacts with A's and U's. And so if we wanted a negative control for our microscale thermophoresis, we used poly-C. It hasn't been shown to interact with nucleolin, and sure enough, when we use this one, we don't see an interaction. So.
Sorry, what are your next steps going to be with this characterization of nucleolin? Like, what are you looking forward to?
Yeah, so the big thing is we are actually sitting down this week and trying to create a plan with our collaborators on what we're doing next. Again, we have kind of these three stages, right? Like I mentioned before, where we have, we don't know how nucleolin is being exported outside of the nucleus, right? We don't know what's mediating this interaction, right? And so this essentially is what is going to be happening with our future studies, right? We want to know what's happening when with how the viral RNA interacts with the capsid, and is nucleolin coordinating that? So like performing FISH experiments, further fluorescence studies, knockdowns on Dr. Laurent's side in Quebec and on our end as well, to try and also figure out, how is nucleolin being exported? So.
Are you going to be going to Quebec to do that work, or are they coming here to work with you, or how's that?
Yeah,
working?
so, well, we mostly just collaborate like virtually, right? And then we send each other data. And yeah, who knows though, with this collaborative partnership, maybe one of us will go there, they'll come here, whichever. Yeah, that we have yet to determine that, but.
No, that's nice. You could get some cross training on some new things that you might not have available here on campus.
Mm-hmm.
What's your favorite part of your project? Sorry.
There's obviously there's a few, but just understanding, like recently also with the G quadruplex and it being able to form a G quadruplex was really interesting. And but like what I'm actually anxious for is to see what is going on with this capsid and viral RNA event with the packaging, right? The capsid protein is it's non-biased towards interactive viral RNA. It's literally just a charge interaction. So, but I'm anxious to see this event right here. And I'm also doing overexpression of several other human proteins that were identified in the pull down. And so it's just cool when things work, seeing that interaction happening, whether it's with microscope thermophoresis or performing other biophysical studies, just being able to mix that human protein with viral RNA and pretty much just a shot in the dark if it's going to happen, and then it works is it's pretty rewarding. I do enjoy that.
Totally.